Welcome to a new episode of Getting Started with Eco-Metabolomics. In this episode, we explain the meaning and importance of metadata in ecological metabolomics studies and experiments. We'll first explain where metadata comes from and why researchers must collect metadata right from the start of an experiment. In the second part, we'll give guidance on correctly organizing and formulating the metadata. What is metadata? Ecological and environmental metadata documents the who, what, when, where, why, and how of data collection for your study. In Eco-Metabolomics, metadata means all information about the study and the Eco-Metabolomics pipeline. The metadata should be complete enough for it to be used by other scientists to replicate the experiment and reproduce its results. Metadata is created during the experimental workflow. It starts with designing the experiment and continues through the collection of samples, sample extraction, mass spectrometric analysis, and data processing. And it's accumulated in the form of various tables and lists. These tables and lists are documents containing information about the samples, the data, and the way the data was acquired, the protocols. They make up the metadata. The metadata is needed for statistical analysis and modeling later on, but also for publishing the study. You can also think of it as a standardized digital lab book that you produce in addition to producing your experimental data. Why is metadata so important? There are several good reasons for high-quality metadata. The most obvious reason is that complete metadata can be reused for submitting raw data to data repositories like metabolites, Redo, and GNPS. The storage of raw data in such repositories is often required by journals as a condition of publication. Therefore, the creation of a metadata file is mandatory at some point anyway. Metadata can, thus, improve your visibility and impact as a researcher because it allows other scientists to discover your work more easily and cite your work more often. And formulating metadata also helps you to follow the guidelines of good scientific practice more easily, helps you contribute to the research community, and to get feedback on your work. Furthermore, metadata is essential for guaranteeing reproducibility and transparency in the scientific community. Data synthesis or meta-analyses and cross-data analyses are not possible if the metadata is bad or absent. How should metadata be organized? Eco-metabolomics is usually done by ecologists and metabolomicists in cooperation. In the course of a metabolomic workflow, the scientists involved create many different lists and documents depending on the step or stage they are at in the workflow. One list from the sampling, another list with the sample weights, an injection list for the instrument, and so on. This doesn't just risk losing information, but it also complicates cooperation between the different operators. As good scientific practice, we recommend creating a metadata file that combines all lists and tables into a single document. A single document including all information and accessible to all collaborators. This document should be in tabular form and used by all collaborators from the beginning of the project. In addition, if you use standardized semantics, computers are able to understand and interpret the metadata, and your data can be more easily discovered by others using search engines. This is machine-readable metadata. In this form, metadata increases transparency between cooperating researchers and leads to greater consistency in the names of species and in sample names and descriptions. This is not only self-explanatory for all cooperating partners, but also saves time and avoids confusion. What should good metadata look like? Metadata consists of information about samples, data, and the methods of data acquisition. The protocols of the different workflow steps can be created separately, and usually they are submitted in or uploaded in text format. The sample and data-related metadata are combined into one table. In this table, the samples are listed row-wise and all categories of information column-wise. With every step of the workflow, more and more information is added, and the table grows in both directions. On the following slides, the basic categories are introduced and explained. Furthermore, we show how the table grows step by step. Please remember that additional categories and columns might be necessary for your personal metadata file. The general part. The sample name. First, samples should have an unambiguous sample name, which is consistent through the whole experiment. Furthermore, don't use numerical code here, because its meaning only becomes clear by looking it up in another table. To make the metadata more easily machine readable, it's best to avoid space characters and special characters in the sample name. The sample type. In addition to the biological samples, there'll also be other sample types in the pipeline. These include processing or solvent blanks, quality controls, and sample pools. The species name. The species name must always appear in the table, even if only one species was used. This information is key for data synthesis and comparisons across studies. To enable community-wide consistency, we recommend using the Latin name of the species used by the NCBI, National Centre of Biotechnology Information, or by GBIF, Global Biodiversity Information Facility. The variant. For the same reasons of consistency, the variant should always be included in the table. Please fill in here the specific accession name, accession number, breeding type, genotypes or mutant, even if your experimental design includes only one variety. Factors or treatments. The metadata category factors and treatments include all values which are fixed by your experimental design and planning. These include the herbivore applied, the part of the plant sampled, plants that are nearby, or the local biodiversity level. Again, please use the NCBI taxonomy or accession number where applicable. If your design doesn't contain any treatment at all, we recommend including one column named treatment anyway and putting control in every row. The traits. The next category, traits, includes all values observed or measured just before or while sampling. These might be plant-specific traits such as the level of herbivory, shoot height, or number of leaves. Or they might be environmental traits like the level of biodiversity observed, soil moisture, or the temperature. As ecological sampling experiments can last for several days and environmental conditions can vary over that period, we also recommend including here the day and time of sampling. It's also possible to include additional data from other analyses. For example, the carbon to nitrogen ratio, if this data is used later for the statistical analyses of the metabolomics data. Technical values. The technical values result from sample preparation and MS analysis. One important value is the sample weight, the amount of ground plant material used for the extraction. This value is usually used for data normalization. Another important technical value is the sample ID, which is in fact the name of the raw data file created by the instrument you used. These IDs consist of the date of analysis, the sample name, the measuring mode, and a serial sequence number. The serial sequence number describes the order of injection. This information should be filled in separate columns to ease filtering and data analysis. The date of analysis and the serial sequence number can be used to test for batch effects. The measuring mode can be used to easily analyze the positive and negative modes separately. Because MS and MS-MS data are different in their structure and also purpose, this information should also be noted in the table. Data processing values. This last category summarizes values resulting from data processing. They include the file name after formatting the raw data from a vendor-specific file format to MZML or MZXML. Please keep in mind that the raw data can only be submitted to data repositories when they are transformed into one of these data formats. Other processing values are metabolomic traits, which are traits derived from processed metabolomics data, like Shannon diversity and chemical richness. These traits can also be included in the statistical analysis. Description and comments. Finally, we highly recommend adding one or several columns for additional comments on individual samples. This information might, for example, include observations made during sample processing. Such observations are highly valuable when it comes to handling outliers in the dataset. The team of EcoMetEoR and ChemBioSys hope this explanation will help you to easily manage your metadata. Thank you for watching.